For the final presentation, we have Etienne Henry. Etienne has been a mission officer for the R&D tech transfer for France BioImaging since 2020, after 11 years as research engineer for CNRS, where he worked on the development of new methodologies based on fluorescent spectroscopy, so fluorescence anisotropy, FRET, fluorescence lifetime imaging, and also single molecule imaging. And he will present together with Luc Lenglet, who has led the Twinkle Factory since its inception in 2018. And Luc is a serial entrepreneur and active mostly in life sciences. For example, with companies such as Dreampore for protein sequencing, Nelogy for sensors. And he is a chemist by training, and as an individual most concerned by biodiversity issues. And he also created a conservatory for hardy orchids. I'm very curious to hear about that if time permits. So both are probably very well suited to talk about technology transfer and how collaborations lead to valorization of research results. And we start with Henri, I believe.
Hello, everyone. Can you hear me well? I think yes.
Yes, we can.
Thank you, Claudia, for giving me the opportunity to present the way in which France BioImaging and research infrastructures in general can help to foster and facilitate research and development and tech transfer. First of all, I would like to make a short reminder about France BioImaging. France BioImaging is the French node of Euro-BioImaging. FBI, it's 24 imaging core facilities, 30 associated teams, more than 300 engineers and researchers, more than 350 setups, 6,000 users, 20 to 25 million of budget per year, 600,000 euros per year to run and develop FBI, and 7 nodes with complementary expertise. The infrastructure is nationally coordinated and distributed with 6 geographical nodes: Paris Center, south of Paris, Bretagne Noire, Bordeaux, Montpellier, and Marseille nodes, and 1 transversal node for image processing and data management. Each node is based on facilities and teams developing methods from chemistry to data processing, instrumentation, biosensors, or sample preparation. FBI is at the crossroad between molecular and cell biology, biophysics and engineering, mathematics and informatics. So France BioImaging primary mission is to develop, promote, and disseminate and provide access to innovative instruments and imaging technologies in the field of bioimaging to scientists. The mission is to foster the technological transfer is at the heart of this mission, and for this, France BioImaging relies on a strong association of leading teams with core facilities. So FBI improve access to advanced technologies and methods, providing technologies expertise in biological imaging, watching with local excellency in scientific topics. FBI give access to commercial or in-house imaging installations of the state-of-the-art, and continuously evaluate strategic plan of equipment, investment, and R&D in the whole territory. FBI promote also training, providing technological expertise to labs and teams, not only on the perimeter of FBI, facilitate infrastructure accessibility, in particular by developing effective teaching training methods for rapidly emerging technologies. And the last but not the least mission is innovation. because FBI bring together research and innovation. FBI speeds up transfer R&D to imaging platforms, promotes new collaboration between R&D labs, and industry, and strengthens attractiveness and competitiveness of our core facilities, institute, labs, and startups. So how to do it or to improve this mission? So when I start my mission for France BioImaging in 220, I identified bottlenecks and leaflets, and based on the feedback from FBI R&D teams, core facilities, users, we have identified issues, and here is some of them. Technology is not enough user-friendly. We have a lack of human resources to stabilize and transfer to facilities. The technology is too specific, not enough user base. We have difficulty to contract with industry through institutional offices for industrial valuation. Concerning image analysis, the instability of open software, economical model, interoperability, large data handling, and the algorithm complexity. And we get some advice from the Scientific Advisory Board, which give us ask us to extend outreach activities, especially targeting specific scientific communities in order to increase the number of external users and understand user needs to ensure that technical developments correspond as closely as possible to the needs. And to tackle these challenges, we set up a series of calls for proposal at the end of 2020. The first season was an internal call to promote the technological transfer from the R&D teams to the facilities by involving, if possible, some startups. We wanted to promote technological transfer from teams to France BioImaging core facilities to facilitate access and service to end users. At start, the technology should be demonstrated in a representative environment, which corresponds to the technology reading level 6. And at the end of the project, the prototype will be usable by the end users until the interpretation of the data, taking care of training, sustainability, and end user autonomy. This call is closed now. But first, I would like to tell you a few words about the funded project. We found 5 projects for this first call, and this project started in September. There are 4 IT projects and 1 instrumental project. The first is IC at FBI, about to broaden the scope of application of IC by implementing several key new bioimage analysis components. The one in Bordeaux is a technological transfer of a single molecule based high content screening platform to the Bordeaux Imaging Center. And Cloudfish is a tool for the analysis of single molecule RNA and DNA fish images. Morphonet, an interactive online morphological browser to explore complex multiscale data. And BioImage IT is an open source framework for integration of image data management with analysis. So we launched a second call this year. For this second call, we would like to extend the outreach of FBI. The idea is to increase the outreach of France BioImaging infrastructure by the development of a new technology by the teams or the core facility of FBI. And this development should meet the needs of users outside the FBI perimeter by connecting the external user needs to technological development inside the teams. And this development should pave the way for the future technological transfer of these prototypes. This call is already open until April 2022 and is open to European community, so don't hesitate to contact us if you are interested in. Within FBI, we have an industrial committee. This committee is composed of a large group like Leica, Zeiss, Nikon, Thermo Fisher, and startups like Twinkle. This tool is used to make the connection between the industrial ecosystem and the academic ecosystems. We want to make the connection between academia and industry to help startups to meet the needs of academia, to make the connection at the core facility level, like the Nikon Imaging Center at Institut Curie, or the loan of equipment. We want to make the connection at the infrastructure level between core facility and everyday teams. And we want to promote emulation, and FBI should be a real breeding ground for startups. And dealing with startups, I now give the floor to Luc Lenglet, which is the CEO of the Twinkle Factory, and who will tell us about the genesis of Twinkle. So Luc, this is yours.
Thank you for the nice introduction, Etienne. So I'm now sharing a presentation of this story that I'm happy to share today with you. The Twinkle Factory is a kind of spin-off of the Ecole normale supérieure and Institut Curie. It was created by three researchers of the ENS and Sorbonne Université, Arnaud Gauthier and Ludovic Julien, who are really developers of new objects, new technologies, and Franck Pérez from Institut Curie in Paris as well, who is really a final user, very active in the cancer research. So Twinkle Biosense was created in 2018 together with PSL University, and I'm the head of Twinkle. I'm not a scientist myself, but used to starting up high-tech companies. And Twinkle is an exclusive worldwide licensee. What is it about? We promote a technology at the Twinkle Factory, which is named FAST, which is a system for labeling proteins by genetic encoding. It's a two-part system with a protein label, which is genetically connected to the protein of interest. It's called FAST. It's not fluorescent itself, and when it is put in the presence of a so-called fluorogen, which is not fluorescent itself either, then it gets fluorescent. What is very unique is that the connection between the tag and the fluorogen is reversible. And this does not exist at all in any other technology. So it's very useful when you want to track a very high speed dynamics inside the cell, protein trafficking, and so on. So far, I would say that this Twinkle Factory is a great dissemination success, meaning that we are not rich yet, but we are very happy with the number of customers worldwide who are implementing the technology. Since the picture that you can see now on your screen, we have added Uruguay, Brazil, Czechia, Spain, and all Scandinavia. And there is one country that you cannot see, which is Singapore, where we have 4 customers. We're very happy that 95% of our customers are abroad, out of France, and we have already 9 of the top 10 of the Shanghai ranking in our customer base. And also, we have an unforeseen success in microbiology. This was not at all foreseen when the researchers started the venture. So, what about FBI in this story? I was not involved at the very beginning and for the nursing phase, I would say, but FBI has been involved from the very nursing step of the Twinkle Factory until now for the scale-up phase, I would say. Why subtle? Because I think that when we started with the France BioImaging, it was not that well structured as described just before by Etienne. But basically, what was the key benefit of this nursing by FBI was definitely to make both ends meet. I mean, the developers and the users, because this is really a key success factor when you want to speed up the dissemination process. And I do agree with the figures that Timo shared just before. It may take years and years before you are ready for the commercial stage. So basically, and to be very specific, what FBI did for us, initially they funded the initial steps. That is to have the equipment available for the team, the development teams were chemists. They were not involved in biology or medical research or whatever. What was very useful also was to gather developers and users through dedicated meetings or general meetings organized by FBI. And a key success factor was to turn researchers into valorization champions. I don't mean to turn researchers into entrepreneurs. This is not the point. The point is to upgrade their mindset, to make them willing to valorize effectively their technology. And what they are most interested in is not willing to make big monies or to build a big company or whatever. It's really to disseminate as fast as possible their technology all across the world, and they're very happy with what we have done so far and our success so far. And also FBI is going to be useful for training core facility users. This is something that we have not done yet because of the virus, but this will get started next year. So again, thank you very much, France BioImaging, for your help at growing the Twinkle Factory.